Thank you very much, Bruce. Just a small correction there. It was actually Miami University of Ohio, so nowhere near as exotic as Miami, Florida. Yes, so thank you very much for the invitation to speak to you today, and I'll be talking about chicken bioreactors for the production of pharmaceutical proteins, and not sorry for the terrible pun in the title. So we all know Dolly, of course, that's why we're here today. And you might be thinking to yourself, well, what does adult somatic cell nuclear transfer in sheep have to do with producing therapeutic proteins in chickens? And if you're thinking not much, you're right. It's actually not Dolly that is of direct application to us, but Polly. Polly followed about a year after Dolly, and along with her sister Molly, they were the first adult SCNT transgenic animals. So not only were they clones, but they also had a transgene inserted into them. And this was a transgene to express human blood clotting factor 9 in her milk. And this was hoped to be a therapeutic protein for human disease. Why would you want to produce a therapeutic protein in an animal in the first place? Well, taking a step back, therapeutic proteins, or biologics they're called, are pretty big business these days. They're very successful in treating what have been historically recalcitrant diseases, particularly chronic illnesses and cancers. And 6 of the 10 best-selling, most profitable drugs in the last year were actually biologics. So biologics are not only big money makers, but they're actually also big money costers. They're very expensive to produce. And the reason for that is that they're typically made in some form of cell culture. They have to be made in a cell because they're a protein. So these are called bioreactors. And when possible, they're made in bacteria because they're the cheapest, or in yeast. But frequently, you can't make proteins in these systems, so you need insect or mammalian cell culture. But all of these systems come with their own problems. So bacteria are not able to produce proteins that require post-translational modifications. They're often incapable of properly folding or having soluble proteins that are desirable. All of these different systems can have yield issues, and mammalian cells in particular are very expensive to run. And the capital cost for these systems, particularly mammalian culture, runs into the hundreds of millions. So ever since we've been able to make transgenic animals, it's been a goal to make animal bioreactors. Animals produce lots of protein naturally, and some of this protein is secreted in an easily accessible form. So for example, lactating animals, and focusing on the sheep, make about 2 kilos of milk per day when they're lactating, of which 6% is protein. So 24 kilos of all the proteins combined if they lactated every day for a year. And the humble chicken, smaller quantities, but a higher proportion of protein in the egg, and still able to produce pretty substantial quantities of protein. So if we could hijack these natural systems and have them drive production of pharmaceutical proteins, that would be pretty great. And this was actually the goal of PPL Therapeutics in combination with the Roslin Institute when the sheep systems were being done. And they would have used chickens, of course, but chickens could not be made transgenic back in the late 80s and early 90s, so they went with sheep. So this is actually Tracy, the transgenic sheep, and I'm ashamed to say I'd never heard of her before I started doing research for this talk. But she was actually created back in 1990 using embryo microinjection. And they inserted the gene for alpha-1 antitrypsin, AAT, into the embryo that produced Tracy. And they got multiple different sheep, but she was the absolute star because 50% of the protein in her milk was AAT, and that's pretty extraordinary. But that was because she had multiple insertions due to the technique used. And these all separated out, and her offspring did not inherit the high expression. And embryo microinjection was really, really inefficient. And so this was what kind of led to the work with nuclear transfer, and the rest is history. But enough about sheep, back to chickens. So chickens can't be cloned. It's just a non-starter. But they can be made transgenic now. And within the last sort of 15 to 20 years, we've managed to make a really efficient system for making transgenic chickens. And chickens have a lot of advantages over mammals for therapeutic protein production. They're a lot faster. You don't have to wait a year to 9 months to get a single calf. Once you've got a founder transgenic, you can have thousands of chickens within a matter of months. They're a lot cheaper to feed and keep and house than cows or goats or sheep. Egg white is an easier source of the protein to purify out the protein, so you don't have to worry about the lipid contents once you've removed the yolk, which is easy to do. And it's a very simple, only about a dozen proteins in eggs, so it's actually quite a simple purification process, it turns out. You have a wider variety of possible targets available to you thanks to the evolutionary distance between chickens and mammals. You can produce proteins that would harm a mammal in the chicken without any effect. So a main example of this that's been shown is erythropoietin, when produced in the mammary glands of mice or rabbits, caused a huge increase in the red blood cells of those animals and harmed their health, whereas mammalian EPO has absolutely no effect in the chicken. And strangely enough, chickens have very similar glycosylation patterns to humans as compared to many types of cell culture and other mammals. And so any proteins that depend on particular glycosylation for their activity actually are more active when made in chickens. And also there's reduced immunogenicity associated with the glycosylation patterns in chickens. So how do we make our transgenic chickens? Well, our main system for making the therapeutic protein versions is a little bit old-fashioned in the days of genome editors now, but we use a lentiviral system. These are replication defective lentiviruses. We put our transgene and promoter of interest into a lentiviral vector, transfect these into cells, which then produce pseudoviral particles. These are injected into the embryo of a newly laid egg, so at about 60,000 cell stage. This is Dr. Adrian Sherman, who does all these injections down behind us in the Greenwood building now. These eggs are then placed into host shells, which are incubated and hatched with the see-through top so that we can watch them develop. The males of these are screened for germline transmission in their semen, crossed with wild-type hens, and the next generation of transgenics will be fully transgenic, not chimeras like these guys. And from there, you can observe your transgene expression. And this was first successfully used in our group with the GFP chickens, which have a ubiquitous promoter and express GFP everywhere and are still a very popular developmental biology model around the world. But for the protein production, we wanted to restrict expression to the oviduct. And ovalbumin is the key promoter for doing this. Ovalbumin is the most abundant protein in egg white, over half the protein in egg white. And as the egg travels down from the top of the oviduct, along layers of egg white get set down, and this is where the protein will be expressed. And then eventually you get the layers of shell, and then it's laid 24 hours later. And we made a modified promoter sequence of 4.4 kilobases, which fits into the lentiviral system and allows for really high expression. In some of our homozygotes, we get up to 3 grams per liter, which is industrially relevant quantities. This is our special bit of kit for purification. So first of all, the egg white has to be clarified and ovomucin precipitated out. This is what gives the egg its jelly-like consistency. We dilute and take that out, and then it's ready to load onto a chromatography column for affinity purification. Most of the other egg white proteins are lost at this stage, and then this gets injected into a size exclusion column, which separates proteins by size. This gets rid of the remaining contaminants from egg white and also any aggregates of our protein that have accumulated. So I'll tell you now about the 3 main targets that we have in our system. The first is interferon alpha, and this was one of our earliest lines using a slightly different version of the promoter that isn't as efficient. But this is a protein that's used in the clinic to treat hepatitis, various forms of cancer, and is also part of the innate immune system fighting infection. So although this doesn't express as well, from one egg we only get about 6 milligrams of protein. It is extremely pure, as you can see by 2 different methods of testing purity, 96 to 98% pure. And activity, so this is a viral plaque assay where cells were infected with influenza A virus and then treated with interferon. And as you can see, this is the green line is a commercially available interferon alpha that's made in E. coli. And these 2 are the egg protein purified by 2 different methods. And the egg protein is able to reduce the titer of virus significantly more than the E. coli produced at the same doses. Our second one is Trastuzumab. This is a monoclonal antibody used to treat HER2 receptor-containing breast cancers. And again, we get a very high purity molecule, 96 to 97%, fully assembled monoclonal antibody. And again, this is a slightly lower amount that we can get at the end from this egg because the system still requires a bit of optimization in terms of getting the transgene exactly right. But that's still plenty for doing in vitro assays. So this is a antibody-dependent cell-mediated cytotoxicity, ADCC assay, and this is thought to be the main mode of action that Trastuzumab uses to kill cancer cells. So what we've got here, MCF cells are low HER2 receptor expressing, SKBR3 are high HER2 receptor expressing. So these patients would not normally receive Trastuzumab, and that's because, as you see here, there's basically no ADCC activity ongoing. Whereas with the high, these are the ones that would receive it. It has a massive increase in the ADCC activity. Now, interestingly, with the egg white, although we do see a sort of 50 to 60% increase in activity with the egg protein compared to the commercially available, the low HER2 expressing also has an enormous uplift. And if this in vitro result were to translate through into the clinic, what this means is you've just increased your patient population that you can treat with this drug. And potentially, you can reduce your dose or have a shorter time course with the more active drug, and therefore make it less expensive for the NHS and patients. Our final protein is CSF1Fc, and this is a novel potential therapeutic with lots of potential applications in all areas of human and animal health. And this is a cytokine dimer, and again, extremely pure, 97 to 98% pure. And this is our star expressor. One egg from one of our lines can give about 50 milligrams of protein. And so in terms of activity, this is a cell line that's dependent on CSF1 for survival. So when you remove CSF1, they die off. When you give them egg CSF1, they survive. And one of the big things that we're interested in with CSF1 is organ regeneration. So this is a ischemia reperfusion injury model where blood flow was cut off to the liver and then it was allowed to reperfuse, and a lot of damage can occur to the organs with this. The sham treated with PBS, you can see that the liver has suffered greatly, but when treated with CSF1, you get hepatocyte regeneration and the liver is very nice and healthy. And this has potential applications in organ transplants, for example. So to summarize, chickens can't be cloned, but they can be genetically modified, and we can do this very efficiently now. Chickens have many advantages over sheep and other mammals for the production of pharmaceutical proteins. And these egg-derived biologics are able to be both highly pure and highly biologically active. So we're now moving towards commercialization of this research. All of these lovely proof of concept studies have shown that we can do this in a viable way at large scale, and so we're now moving towards commercializing our technology. But we're also always interested in collaboration, so if you have any interesting proteins that you would like us to make in chickens, please come and talk to me. So these, again, a big team of people who have contributed to all of this work, and in particular, the animal staff at the Greenwood building, who we wouldn't be able to do any of it if they weren't there looking after our chickens. And thank you very much for your attention.
I have reread my crib sheet, and it's my mistake. Apologies to your undergraduate at Ohio. Any questions for Lissa?
I was curious whether there are any regulatory considerations for proteins derived from livestock compared to recombinant proteins.
Yep, so there are 3 drugs already on the market from transgenic animals, including one from chickens. The regulatory approach is that they regulate the entire animal. They look after, so the animal is treated as the feedstock or the master cell line or whatever. So it's the animal that they look at for the genetic modification. They test for diseases and that sort of thing. But the regulators do seem to be on board now, and so you have things in the human clinical space which are fully approved and on the market and have been since 2009.
And an unrelated question. So you mentioned lentiviral gene delivery, which has obvious limitations. Is it possible to use genome editing as well in chicken eggs?
So that's technology is a bit further behind in the chickens than in the larger animals, but you can do genome editing in primordial germ cells of birds, which can then be injected into embryos and lead to transgenics in the same way, in a slightly different way than the lentivirus, but it is possible to edit them, and we've got groups in the Roslin doing that. In terms of inserting a transgene, it's still faster and easier to do the lentivirus at the moment, and most of the proteins that you would want to make have a small enough sequence that it fits into the lentivirus. But it's certainly something that we're looking into for improving things and making it potentially more regulator friendly as well, of being able to control exactly where the gene goes in.
Do we have another question? We do down here.
Hi. It's quite interesting that now we have Synagiva drug that is approved. How different is your system from Synagiva? Because the drug is for lysosomal, so the delivery to lysosomal, it's better than other systems that we use in clinic for lysosomal disorders. So it's a really nice turn that we are having a drug with a different system, but how is different your system related to Synagiva?
It's actually pretty much the same. They also use a virus to make their transgenics, and then they purify the protein out of their eggs, and it's basically the same. Yeah.
You said that chickens can't be cloned, so is this a technical barrier or is it more conceptual?
Technical. The embryo is in an enormous yolk. Cutting open the chickens and trying to transfer nuclei, it just doesn't work. And it's not really worth it because the efficiency is very, very poor. Anyway, even if we could succeed.
You mentioned that your protein Trastuzumab 021 is more effective than the one which has been produced previously in mammalian cells. So what could be the reason for that?
We're almost certain it's the glycosylation because Herceptin is produced in CHO cells, which is rodent. And we think that the glycosylation of the human antibody with the chicken glycosylation is the main function. And this has already actually kind of been played with in the industrial space already, of changing the glycosylation patterns on monoclonals.
Any further questions for Lissa? One more down here.
Just wondering about these monoclonal antibodies. So I have Herceptin or any anti-cancer monoclonal antibodies, usually a huge market. So how many chickens do you think you need to produce enough for what is needed in the market?
That's a really good question. For those, it's really hard to say because our current system is not efficient enough for monoclonal production, and it's partly to do with the way the transgene is constructed. It uses a 2A peptide, and the one that we have is the oldest and least efficient version. So we think that we're actually losing quite a lot that's not assembling properly. So we need to make some changes to that. But the actual amount of protein that is being, that is detected by quantitative Western in the monoclonal chicken is in the 1.5 grams per liter range. So if we could get 90% of that as properly assembled antibody, then that's that, and then have a homozygote, so there's 3 grams per liter, then you suddenly need a lot fewer chickens. But it really depends on the dose, and my understanding is that the dose can be quite variable depending on the particular patient population, what exactly they're trying to do, which stage of chemotherapy. So it's very, very hard to answer that question. Thousands to hundreds of thousands, but a farm of hundreds of thousands of chickens is actually already exists, and the production mechanisms for taking hundreds of thousands of chicken eggs and processing them already exists industrially, both in the food industry and in the farm industry for vaccines. So it would just be the time frame of getting all your chickens in the shed.
Any further? We've got a hand here in the middle.
Very nice talk. Can I ask what the levels of myristoylation and other cross-linking, because that's really important in some of the recombinant proteins, and to actually make them, work in humans. Is that something that you've looked at?
That. It's a bit outside of our particular area of expertise, but that's something that we will be looking to have analysed externally. Yeah.
With that, I think we will thank Lissa again for an excellent talk.